All right, so I'm arriving to Dr. Grimmer's place now. 26 Strasse. Yep. Kniser, Technical University of Munich. So, let's go inside and have our interview.
I'm Jesse Silvera with Spirit of the Coast Analytics. Today we're going to be with Dr. Timo Grimmer, and we're here in Munich, Germany to discuss the Alzheimer's space and Anavex Life Sciences Corporation. Dr. Grimmer is a board-certified psychiatrist and psychotherapist, an associate professor working as a specialist registrar at the Department of Psychiatry and Psychotherapy, Technical University of Munich, School of Medicine, Munich, Germany. So I want to thank you today, sir, for having me for this interview. It's a pleasure to meet you and visit the fine city of Munich, which I was exploring all yesterday and earlier today. I was hoping that we could start off with your role with Anavex. I was wondering if you could elaborate on where your connection with the company began and where you are now with Anavex, perhaps a little about your current roles and responsibilities as well, anything that you have to say.
Yeah. Thank you so much, and again, thank you for being here and taking you the time. My Anavex connection is, I think, several years ago. I know a few of the persons working for the companies for longer times, and they introduced to me their compound and the mode of action, which I found very fascinating because I need to admit I never heard about this mechanism before, and this despite studying medicine before. So this was surprising for me, something new to learn. And then I got the impression, this is really exciting. This could do something, an absolutely new mechanism of action on the one hand, and of course on the other hand, it could be the case that it does nothing at all, and it is not an important mechanism. But I followed this closely, and the company asked me to serve as an investigator and as the national coordinating investigator here in Germany for their phase 2, phase 3 trial with 273, I think the compound name is 273. And and now great news. The company has managed to finish the study, which was not absolutely true. I think at the beginning they had some recruitment problems, and so I think this was the major reason to open it to other countries like my country. And but then apparently everything worked smooth and fine. And now last December, seeing the data set for the first time, which I found exciting to see that there's obviously a clinical signal in it. But staying with the clinical signal, they apparently see clinical benefit, which is good. So we have treatments, drug treatments for Alzheimer's disease at the stage of dementia. The cholinesterase inhibitors, memantine, which have some modest effect, so they also improve symptomatology. But of course, the effect sizes are limited, and everything else that could help to decrease the burden to patients would be highly appreciated. And still not seen every single analysis on the clinical data, but staying to what have been disclosed at CTAD, for example, this looks very promising. So it appears that patients improve. So consistent with the idea that it is really doing some symptomatic relief. So which is exciting. First, this mechanism really does something helpful in Alzheimer's disease brain. And of course, it is very needed for patients that already suffering from symptoms and cognitive impairment and impairment in their activities of daily living. So everything that can relieve this burden is, I think, intuitively associated with an increase in quality of life for the patient, with a decrease of burden to carers and so on. And so very promising so far.
Isn't it true that you are now an advisor for Anavex?
Another aspect, yeah. And then they asked me just recently whether I like to join their scientific advisory board, which I feel very honored to be asked. And based on the fact that this is such an exciting mechanism, I'm gladly accepted this.
Great. Okay. So as you mentioned, you're principal investigator for the German population for the trial, right? So what was your access to patients like during the Alzheimer's trial? Were you seeing any of the patients that were on the drug, or were you simply overseeing some of the clinical sites like that?
The latter. But this was not we had some administrative issues, so the trial was initiated on my side much later than anticipated. So unfortunately, when my side was really ready to recruit patients, the sample was already almost recruited. So it did not make sense to in this few weeks which left to include patients, which is a pity from the perspective now. But hopefully, the company will do additional studies, or if all their hope come true, maybe already receive some kind of approval also, so that the access to this drug might be able to my patients too. Hopefully,
that'd be awesome.
Mm.
With that said, it sounds like the answer is going to be no. But are you familiar with, have you heard of any real world evidence? I'm not sure if you can even speak about it, but maybe patients' behavior improving from the caregiver or the patients themselves or any of the clinicians. Is that something that do you have insights into?
Yeah, yes and no, but in general, I've learned to be very reluctant to believe anything that I'm learning from the patients and caregivers' perspective with regards to, this drug is helpful and I see improvement and benefit, because there's what we call in medicine is this concept of a placebo effect, which I don't think is not the best word one could find. The idea that we are just doing something we believe is good for a patient, and the patient and the carers think we are doing something and participating in something that is good. This is relieving and is associated with improvement for everyone. I think this is the main idea and concept of medicine at all. But in a drug trial, of course, is what we then see that patients do improve, or at least subjectively improve, or carers notice improvement, which could be due to the intervention, could also be just being based on the fact that they're in the trial and have put so much hope in it. So me personally doing drug trials for around 25 years now, and what me and my teams always have done at the end, before the unblinding occurred, saying, just betting on the cases and say who's been on active drug, who's been on placebo. And to be honest, it almost failed every time to predict who was really, regardless whether the drug worked at the end or the trial was positive or negative. But the individual fluctuation and variability between patients and the course of the Alzheimer's disease between patients is so large, it is almost impossible. So I'm very reluctant if, for example, a patient comes to me and say, I've done whatever intervention and now I get better, so probably this is associated. It might be due to the fact that the intervention was causative for this improvement, but it could also be by chance. So I'm relying on the statistical analysis, on the data set, on the sample to be more robust. And if you look at the data, of course, it appears that patients in both groups, patients have declined cognitively and in activities and daily living. And in both groups, patients have improved on cognition and global scales and whatever. And at the end, it's only the percentage rate that we can really rely on and saying, it appears that those on drug have, as a sum or as a sample, the better outcome.
Oh, it's fantastic.
One could ask that, is it possible to predict the course or the individual course of a patient, and that the variability of individual courses in Alzheimer's disease is much higher than, for example, if you're only looking at motor function in Parkinson's disease. I think if I have not seen this publication or the manuscript you're referring to, but my assumption would be that if you're only looking to a small part, so for example, motor function in Parkinson's disease, then I believe that this is much more predictable, because it relies to some cells or some plaques of neurons in the midbrain, and if they are diseased and they are degenerating, this can be easily predicted. But as more complex processes you're focusing, like memory or activities of daily living, which require large networks of brain cells, of course it makes it much more difficult to predict at what time point a particular function might got impaired. So this is the idea that if you need hundreds of neurons around the brain to maintain a function, then if one of these essential ones does not work anymore, then on the symptoms, this particular symptom will then occur. And so it's very difficult to predict, based on Alzheimer's disease with a very diffuse pathology, when this particular moment will appear. And another aspect is, as more complex brain functions are, the more different they are coded, or they in an individual brain. So even if you're, for example, considering to set a or to destroy certain neurons stereotactically, for example, in the brain of two different patients, you cannot predict whether both patients will then suffer from the same symptoms.
Right.
So I think this makes it more complicated and much more difficult to predict. And of course, this is one of the reasons why everyone is so keen and so glad that we now have the opportunity to have specific biomarkers to track AD pathology, because this makes it, of course, for example, in drug development processes, much more easy to predict, may a drug work, and does not require such large sample sizes, which of course you need to have if you like to show the clinical benefit, which of course is the prerequisite to approve a drug that should help patients. But if you're during the process of developing drugs, of course these biomarkers are tremendously helpful.
It's harder to tell exactly when Alzheimer's patients are going to degrade and what exactly is causing that, et cetera. So that really helps me out. And we will talk about biomarkers a little bit later as well. Thank you for bringing that up.
Yeah, but from this biomarker perspective, we can see that then Alzheimer's disease appears to be very comparable between patients. So it is not the case that there's so much variability in the speed of the real disease progress, but of course on the symptoms level, it is much more difficult to presume, based on the fact it works and suddenly a network breaks down and a symptom occurs. And this is also from patient perspective very difficult to us, or to explain. They say, "Oh, I've been so stable so long, and now just recently, 2 weeks ago, it speeded up," because then they realized that very important ability does not work anymore, for example.
Okay, thank you. So I'd like to go to our next question, which is Anavex's drug, known as blarcamazine, is normally described as a sigma-1 agonist, but it also features muscarinic 1, 2, 3, and 4 agonism, as well as NDMAR, N-methyl-D-asparitic acid, and sodium channel site 2 agonism. Do you think some of those other binding affinities are providing substantial benefit to the patient cognition and behavior? Is that something that you are familiar with, the other binding affinities of blarcamazine?
Yes, I've read all of these, and I find it very difficult. So, short answer, I have no idea. Longer answer is it could be, and precise answer is it is it appeared to me that it is very hard really to predict from the clinical effects based on receptor binding or whatever strategy, at least in psychiatric and neurological disorders and their treatments. So we have pharmacological treatments where we know very well what they're doing on a cell basis, but have still difficulties to understand why this shows this specific clinical effect at the end. So I'm very reluctant. I love this idea to generate hypotheses and saying, yeah, this should work or could work for it. But if it really does, and what really will be the result on the symptom level, on the clinical level at the end, is very hard to predict. And I believe the only opportunity we have is to run clinical trials then, and just to see whether what we're hypothetically have found out does really translate in some clinically meaningful impact. So, yeah, I have no idea which of the mechanisms is really doing which part of all of this, but I think I'm based on the fact that I'm a clinician scientist. Maybe I'm not the best person to talk to about this.
No, that's perfect. Thank you. So I do want to shift gears a little bit here. I read on one of your abstracts titled "The Cost of Early Diagnosis of Cognitive Decline in German Memory Clinics." This was published in 2022. Within the abstract, you and the other authors quote the cost of an amyloid PET scan costs nearly 5,000 euros. Considering the push for lecanemab approval by the FDA recently, and now potentially abroad with the EMA, can you expand upon your thoughts on the price of Leqembi at about $26,000 annually, plus additional cost of amyloid monitoring, and how you see those costs fitting into the European healthcare model? So do you believe that lecanemab, or Leqembi as it's now known, do you believe it's affordable to Europeans? What about Germans specifically? And is there any context you can provide from a cost burden perspective? Anything that you have on that would be appreciated.
Very interesting question. So I think the lecanemab is could be the first drug with the disease modifying properties that could get approved in Europe and then in my country first. And if it would be, of course, this would be very great things, because we have nothing to slow down the course of Alzheimer's disease. And I believe that this anti-amyloid mechanism of action is a very important one, and that this really alters the course. And I also believe that, of course, in this clinical trial, this phase 3 trial with lecanemab, this, of course, was not tried to assess the maximal benefit of it. It was a trial to prove that it is working at all. So the main idea is, of course, if we, in easy words here, what we hypothesize is that amyloid is toxic, and if we remove the toxic agent, this should be beneficial to the brain and to patients. And but what we have done, or what the company has done, to run an experiment, doing one experiment with twofold or two aims in one, saying get rid of the toxic agent and expect the patients to stabilize. I think the better experiment, or the best experiment, of course, would have been to see, saying, first get rid of the toxic agent and then do a second experiment and see how patients stabilize. This would be the perfect experiment to see what the effect size or the expected added value of this drug is. Yeah, the maximal added value is. This said first, but your question was more to costs, of course. Yeah, this is a monoclonal antibody, very expensive to synthesize. And as you already mentioned, it would be a very effortful treatment strategy, which means patients need to show up to physician's office every 2 weeks. need to get an infusion, need to undergo MRI assessment. As you mentioned, need an amyloid positivity proof, so maybe a lumbar puncture or an amyloid PET. So all of them, this is costful and expensive. I think I hope that it's still true that I'm living in a very rich country and that we still consider us to be part of what we call first world. So hopefully I'm living in a system that can afford to reimburse efficacious therapy. On the one hand, on the other hand, I understand this. Yeah, you mentioned numbers, so if one would just thinking about something, 20,000 euros or what you mentioned per year, and then multiply this by the numbers of potential patients, I also can see this is a very high number that comes out and makes it very difficult for the payers to reimburse for this. Very difficult decision. I don't think that Alzheimer's disease is a condition that is not worth to be treated. And I do not see any difference why to treat a patient with dementia or not to treat a patient with dementia, but to treat a patient with cancer, for example. And we have cancer therapy that are very costful, and we have therapies for other diseases which are even more costful. So I see the struggle the society will be in, but at the end, I hope that we can still afford, we as a German society, to reimburse for efficacious treatment, and this will also apply to Alzheimer's disease patients, and that but we will see how the situation evolves. Of course, we have, as you know, we have a clear mechanism to evaluate what is called this additional benefit for a new treatment, and if this analysis is positive at the end, I'm very confident that this will be reimbursed.
Okay. No. Oh, that's great. Thank you. I think that kind of along those lines is considering the EMA's Alzheimer's guidance and multiplicity guidance from 2018. Do you believe Anavex's 2B3 design was suitable in addressing this instruction? So do you believe that the way that they set up their trial design is conducive to being approved in Europe, for example? Their co-primary endpoints featuring a cognitive and functional endpoint and a global score as a secondary endpoint. Trends between the FDA and EMA indicate that European regulators favor efficacy and safety over the FDA. So it would seem to me that the EMA is more reasonable when it comes to wanting a drug that is efficacious, safe, cost efficient, stuff like that. So would you say that blarcamazine or the 2B3 trial ran by Anavex kind of fits or checks those boxes that the EMA is kind of looking for when it comes to drug approval?
Yeah. So another interesting question. I'm not a member of the EMA first, so
Of course.
I think I'm allowed to say that I've been invited to consult for them, but of course this does not. I do not want to run into their assessment process, but in principle, I think the endpoints are acceptable. Yeah, this is in accordance with what has been written by EMA and FDA, saying, of course, we need, if you're looking at cognition, we need to look at another so-called more important domain, which is function. And I would agree that, of course, that if I personally would suffer from dementia, at the end, I do not care how good my memory is, but I do care how independent I can live my life, regardless whatever is being done in a cognitive test. So I'm absolutely in line with their proposals, and I think that the phase 2b/3 design does cover all this. They use standard assessments, which are usually accepted by these authorities and also requested by these authorities to be used. So I think this box can be checked. And then if it goes to approval, of course, this depends and this varies. to my understanding, the main concept is to see we love to see a very robust effect of a drug in a randomized controlled trial, and we like to see this independently in a second trial too. This is so what is usually being considered or being wished for and expected from the EMA. Of course, there are exceptions. Then, for example, if you go to rare diseases, then this exception can be saying it's so difficult to find any patients and be so happy if you manage to run one trial. And of course, there are ways to deal with it. And of course, there are ways to say, so they're kind of, to some extent, comparable concept like the accelerated approval pathway that the FDA has, which is this conditional market approval that EMA has. So there would be pathways one could consider to go forward with a single trial. But to my typical expectation would be that EMA would say this is a very interesting trial, and just personally speculating, no precise idea what EMA would really say, but saying this looks very convincing to us. We suggest to you run a second trial, a phase 3 trial, and if you can replicate this result, then probably, we are very happy to, and of course, if safety is acceptable and so on, we're happy to approve this trial.
Great, thank you.
That's just personally,
Yeah.
I like to share. I've learned about the company's hopes, of course, saying, we've seen such good signals, and so I think there will be an exchange in discussion with the regulators and saying whether someone says, agrees to them and saying that this looks so exciting, we already make this accessible to patients, which of course is another perspective I can I find very important to saying, of course there are suffering patients on the other hand who are seeking new, better treatments, and this is this dilemma, so to say, what is the best structure in order to be sure that you're only approving drugs that are really helpful and not just based by chance in a single trial. And on the other hand, of course, not to prevent patients to be treated who desperately need treatment. A difficult situation. I'm so very lucky that I'm not part of the regulated boards, but I find it very interesting discussions always with them, and I think they do very thorough assessments. And one can look, for example, at the aducanumab story, where I think the European regulators have very clearly said, saying, of course, you have we see that there is one positive phase 3 trial, but we also see that you have a negative phase 3 trial. So, and they suggested run a new trial and make it more convincing. And I've seen the more, exploratory approach of the FDA, saying, we do sub-analysis on our own. We like to find out what this drug can do, and so it's a more investigational approach of saying, okay, we are already convinced, and doing some kind of conditional approval or accelerated approval for this drug. And I think both approaches are absolutely understandable and acceptable.
Perfect. I guess I'll just make a comment, not so much a question, is I kind of think that as a part of their regulatory package, Anavex would probably include the phase 2 Parkinson's disease dementia trial data that they had. The outcomes were obviously quite efficacious, especially in, surprised me, the motor improvement on UPDRS was double the clinically meaningful benefit, and the episodic memory, which has obviously strong correlation with cognition in Alzheimer's patients as well, was very high. So while it's not another Alzheimer's trial, I do think that they'll probably try rolling that up in the regulatory package.
Mm-hmm.
And I did have a question, kind of more so, I think this is probably right up your alley, is at least in the investor community, there's been a lot of turmoil, we'll say, over the use of odds ratio. And I was just wondering, can you comment on the use of odds ratio and benefits of using odd ratio for clinical trial data reads? So just anything that you have to comment on odds ratio being used.
Yes, of course, it is a standard measure how we in medicine, in clinical medicine, assess the effect sizes or the predicted effect sizes of a certain intervention. So this is of course common use. With regards to drug development and drug trials, I've seen this rarely being reported so far. So I found this surprising to me, to seeing this at the CTAD presentation, but understandable from the idea saying, yeah, we would really like to see who, or to show and disclose the potential effect of showing a clinically meaningful benefit. I believe that if the study is published, that the whole data set will include all these traditional analysis one is more familiar with. But I found it surprising at the beginning, saying, yeah, why am I not seeing these usual curves here? But I understood the point to saying, yeah, we would like to make the point that there's a clinically meaningful effect and make this more clear. So I think it could be comparable to, or at least being discussed with colleagues, and all say, yeah, this is fascinating, but it's a little unusual presentation mode. And of course, everyone looks forward to seeing the whole data set disclosed at further meetings or in a publication.
Perfect, thank you. During the management conference call that you attended with Dr. George Perry in December, you were hesitant to say that Anavex's 2B3 trial was pivotal, that it could garner regulatory approval, as they say. Considering lecanemab's accelerated approval by the FDA on 6 January, which was a month after the management call, they got that approved the phase 2 trial data alone. Would you say that your position has changed at all on that? I think that you kind of already addressed this earlier. And again, Dr. Mixon sure seems confident that the trial is pivotal. You did already kind of hit on this, but is there anything else that you'd like to kind of mention now knowing that lecanemab was approved after that conference call in phase 2 data alone?
Yes, I think this is the same idea that I want not to mix into the business of the regulators. So being very careful in how to flag trial data. So I would love to stay with my personal assessment of the data and being very careful in order not to do anyone's else business. And if regulators, I would love and I would congratulate Anavex if this trial would be considered pivotal, but I'm not the one flagging it as being so. And you're mentioning that for example, this lecanemab accelerated approval, it was an interesting time point, I thought, when this decision has been made. Yeah, so that at least by coincidence, the phase 3 trial data of lecanemab has been disclosed to public before this accelerated approval has been given based on the phase 2 trial data, which does not need to have anything, could be just, does not need to be an association between these two facts. But for me, as thinking I would be part of a regulating agency, of course, I would be more confident to grant an accelerated approval based on the phase 2 trial data set if I have already seen the phase 3 trial data set, regardless that I cannot formally take this into account for the approval process. But just my personal assumption.
That makes a lot of sense. And going back to one of my comments from earlier, I think that that's one of the reasons that I personally have hoped that this is a pivotal phase trial for Anavex because of their success in their Parkinson's disease dementia phase 2 trial, as well as it's pretty different, but their Rett Syndrome efficacy as well. I think that there's a lot of reason to believe that blarcamesine is helpful across CNS for most disorders, and for that reason, I think, like you mentioned, while maybe they're not using the Rett Syndrome data during their approval process, it may come in as like they are being successful in multiple indications across the board, and that may just come as a factor. But that's just a comment that I have.
Yeah, it's very fascinating, and I'm very curious to see where they can see effects in which CNS diseases, which is very astonishing to me. Yeah, so based on the fact, as pointed out to earlier, that I've not even been aware of this mechanism of action several years ago, and I'm now seeing that it appears to have an impact in so different disease processes. This is the major part of my excitement.
Me as well. So speaking of lecanemab, and you kind of were talking about biomarkers a little bit earlier, but speaking of lecanemab, it was mentioned at CTAD, which you were present at, right?
Yeah.
I was also there. Didn't see you, but I was sitting in the front row during Dr. McFarland's presentation. But so speaking of lecanemab, it was mentioned at CTAD 2022 that when patients were titrated off of the drug, amyloid began to return in the brain. And considering the definition of disease modification by the FDA and the EMA, and again, in just your opinion, would you or would you not say that the return of amyloid proves or indicates that lecanemab does not create a persistent modification in pathology or course of disease progression? Or any thoughts that you have on that would be great. So amyloid's returning after you stop taking the drug. So is the drug really causing or creating a persistent effect?
Yeah, I think there is a persistent effect. Then the idea of disease modification is not, to my understanding, that it needs to be a cure or to stop anything or really to heal a disease, but to alter the course. So the idea is, if those patients who've been put on the drug, getting rid of amyloid, should have a better clinical course, and if you then, for example, start to titrate the placebo group and try to see whether they can catch up, this should not be the case. They should not catch up because they've been started 2 years later, and so the disease had 2 years additional time to do harmful effects to the brain, so they never will catch up again. I don't think it is the idea of the concept of disease modification is meant with regards that the disease is not allowed to reoccur if you stop treatment. I think this is normal. This is what we expect. Yeah? It is the mode of action is to remove a toxic agent, but you're not healing the underlying cause why this toxic agent develops in the brain. So if you stop treatment, of course, it comes back. But what you have altered during this time this time free of toxic agents in the brain, at that time, disease does not progress, or at least progress at a slower rate. And this is the idea of altering the course of the disease and of disease modification, to my understanding. So in short, the answer is no.
Okay, thank you very much. So out of all current therapeutics or upcoming therapeutics that I may not be aware of, are there any that you see that would be a decent combination with blarcamesine? It seems like all of the medical community as a whole is thinking that for Alzheimer's treatment, there needs to be a combination therapy. So is there any drugs that you see that kind of jive well with Anavex's MOA, or any gaps that blarcamesine has that you think would be filled by a different drug, perhaps?
Yeah, so very important point there. Considering me as an expert in Alzheimer's disease, of course, I need to include the statement that the future of Alzheimer's disease treatment is probably a combination therapy. Yeah, this is a sentence I love to say since 20 years. But I would love to have compounds for this combinations therapy first. So the only reasonable combination to think about is, of course, is it worth to combine an anti-amyloid monoclonal antibody with this 273 compound? And my short answer is probably yes. Of course, we need to do some safety data, of course, to rule out that any interaction I cannot imagine, but that they are not present, because it's not prophecy, it's medicine what we're doing. So we can do an experiment in order to rule out that any interactions occur between these two mode of actions. But I could foresee that it is very helpful because in fact these monoclonal antibodies will not relieve any patient from any symptoms. They just slow down, they slow down the progression, but which will mean if I'm already a demented person and have a burden and being impaired and relying on help, this will not get better by a monoclonal antibody. Of course, I love the idea that I will not get worse or worse at a slower rate than without the treatment. But of course, I would love to be being put on a treatment that improves my symptomatology. So this absolutely makes sense, and is compared to the concept that is run with the monoclonal antibodies to put everyone on a background therapy with the antidementic drugs that are already approved, and then in addition trying to show or aiming to show that you can see an additional benefit on top. And so I think if we have an additional player with 273, of course, one could combine this. And of course, one could speculate with other not approved treatment regimens and to combine them, but I don't think this is reasonable at the current stage.
Sure.
Or at least I'm being reluctant doing so because first I need to have this one compound approved before speculating on combining it with another one.
Of course. All right, thank you so much for the context there. So going back to some of the publications that you have authored or co-authored over the years, it does seem that as I'm sure you are aware, you have quite the expertise when it comes to cognitive biomarkers, especially those pertaining to Alzheimer's and frontotemporal dementia. What would you say is the current atmosphere with cognitive biomarkers? They seem to be good for identifying chances of eventually getting Alzheimer's, even many years in advance. It would seem 10, 20 years in advance. But what about their utility during clinical trials? Do you have anything else that you'd like to talk about with biomarkers and their use during clinical trials?
Yeah, I think this is a very rapidly evolving field. Yeah, so for example, several years ago, 5 years ago, we've been so glad if we could manage to do some serial lumbar punctures and doing a CSF assessment on the long run and have any biomarker data available. Then the PET ligands came up, which allows us to specifically target all parts of AD pathology like amyloid tau and, for example, using FDG PET or neurodegeneration too, making it much more interesting. We had the idea to track atrophy with MRI, and now we're starting to understand what can be done in easier to obtain body fluids like blood.
Yeah.
And the field is so rapidly evolving. I have the difficulties to catch up with everything important, new data set and new test that is coming out currently. So I believe that this. Yeah, and what have we seen? Of course, it has helped us a lot in understanding mode of actions very early and helping us to objectively track such a disease, which could on a subjective level have such fluctuations in course and making it so difficult to do comparisons. So I love to see them included in clinical trials, and we're still in the learning phase. Yeah. So you mentioned lecanemab. They have shown, based on the fact that their open label extension study for their phase 2 trial population took some time to get approved. So persons have been there have been a gap between this double-blind period and this open label extension, depending on how early the patients have finished the double-blind phase, which means in fact there have been some interval, and in this interval, the company has conducted blood draws, and we are now learning to see how, as you mentioned, how these biomarkers already re-evolve in what time, and I think we will learn a lot about things like frequency, when can we stop treatment, when to switch to maintenance therapy, and so on. I'm also very excited on all these developments we have here. And as you mentioned, probably their main target or their main use will be in clinical drug development. And of course, one could speculate a long time on their usability and the impact of doing this for diagnostic purposes, as you're mentioning. One could theoretically probably do some kind of fairly reliable Alzheimer's disease diagnostic 10, 15 years before symptom onset, with all the ethical implications, saying if it's not associated with any treatment, then of course, why should we do so? We would only introduce anxiety, fear to apparently healthy persons, and saying, you're feeling healthy, but you're already developing a progressive disease. But on the other hand, of course, if we get considering this idea of disease modification, it would absolutely make sense in a trial design if we could offer some trial participation and saying, we are not now knowing that you have pathology in the brain, but we can offer you an intervention to alter your fate, and then of course the question would come up, saying the majority of patients with Alzheimer's disease are elderly persons, so I could absolutely agree to do a diagnostic test for Alzheimer's disease in for example, 50-year-old person, saying if I can predict you will develop symptoms at the age of 60 or so, the probability is very high that the patient is still living at this age. If it turns scenario to saying doing this in a cognitively healthy 80-year-old person, and I can say, now we see a pathology you probably will suffer from in 5 to 10 years. The patient say, thank you, doctor, that you're so confident that I'm still living in 10 years. So it could be very difficult, and at the end a very difficult debate. And I'm hoping that the companies developing their diagnostic tests will stay in close contact with researchers and clinicians like myself, as they already do, which I would love to stay in exchange and see what would be the appropriate use for such tests in the future.
I'm really excited for the blood diagnostic tests. It seems like they're almost as accurate as the others, and way less or way more cost efficient. If you had to choose maybe a few biomarkers, let's say there's neurogranin-based one, tau, amyloid, stuff like that. What would you say is like the most accurate biomarker in your opinion? Is that something that you have an opinion on? For Alzheimer's and then for frontotemporal dementia.
It's a very difficult question. So currently we are still we're still in the learning process. So at the beginning we thought that the phosphorylated tau peptide with the phosphorylation site at 181 is a perfect marker for tau pathology. So I think it was a CTAT, saying a bunch of other phosphorylated species, and they've been correlated with the signal of amyloid PET, the signal of tau PET, and saying it depends. They are not such a clear tau marker than being initially thought. So this first year was still in the learning process. Second is, of course, it depends on the question that you're raising. So if the idea is you need to prove whom to put on an anti-amyloid treatment, for example, anti-amyloid monoclonal, we're still in the learning process first. The second is, it depends on the question you'd like to answer. So if the question is, whom to put on an anti-amyloid treatment, of course, you need an amyloid marker and nothing else. If you like to do a diagnosis of Alzheimer's disease, one would love to have one being a very good surrogate for amyloid pathology and tau pathology in the brain. If you're thinking about you need a marker to decide on when to stop, for example, an anti-amyloid treatment strategy, one could thinking about, I would love to see a marker of neurodegeneration. If it's going down, then it may be a good point to stop treatment, and then one could considering which would be the best thing we love to see, or which marker we love to see going up again in order to decide to reintroduce the drug. So it would depend on the scenario at the end. And then this first and third point is, of course, it's more associated with accuracy and robustness. So how easily one could do this test, how accurate is the result. So we have certain tests that can still require, for example, expensive techniques like mass spec technology, which makes it complicated because you need to send it to a very specialized lab. It makes it probably not very cheap at the end, which would preclude using it for large patient screening and so on. So I think I'm not in the position yet to clearly predict which one I would love to have and which one would be best for me, because of all the said before.
Okay. It seems like Anavex is trying to make sigma-1 expression and occupancy as a biomarker. Do you have any opinions on that?
I think it would be a very interesting marker to see, for example, whether one could imagine that there's, or with every drug, there are responders and non-responders, for example. And so one could imagine that such tests could be helpful in order to assess, or maybe it is helpful then to identify which patients might profit best on such a new treatment. It could, in principle, serve to identify persons who are the right candidates to be put on the drug. And so I think it makes sense if you have a new mechanism of action to seek for biomarkers which are able to directly measure this one in order to see how the situation changed during therapy. One could imagine that during the course of Alzheimer's disease, the system is getting abnormal at a certain time point, and maybe this would then be helpful to identify at which time point during the course of the disease the best time for an intervention is, and so on. So one could learn a lot. So I think it's a very good initiative, and if I see data, I can comment on for what it is useful.
So hypothetically speaking, if sigma-1 wild type is in fact tied directly to efficacy, I just think that's very exciting for Germany and Europeans because according to wide range meta-analysis of genomics, it seems like Europeans have by quite a margin the highest sigma-1 wild type percentage. So I think that's really an important metric to consider. I want to thank you again for your time today and the time of my viewers. And there's just two more questions and we can close up. Are you excited about any of Anavex's other indications? How about Anavex 371? And they're planning on running a frontotemporal dementia trial in that. Do you have any thoughts about their other indications?
Yeah, based on the fact that they have seen some promising signal in their PD dementia trial, seen a very promising signal in their AD trial, I would, if someone asked me, I would encourage them to go in as many as of these diseases. And of course, you're mentioning, for example, frontotemporal dementia, in which we have no treatment at all available. So of course, I think that the unmet medical need couldn't be higher than in such diseases. And we have here, my colleagues and I have what we call some kind of or for traditional, no, it's the wrong way to address this. I think we have a focus on frontotemporal dementia here. So fortunately, neurologists and psychiatrists are referring their presumed cases to us. which gives us the ability to see more patients and in principle run better trials here. But in fact, we do not have anything on the drug side in order to decrease the burden of their disease and of their symptoms. I would love to see if this could be helpful there. And another problem we have in frontotemporal dementia is that the underlying etiology is very heterogeneous. So clear difference to Alzheimer's disease or Parkinson's disease. So several kind of protein deficits can cause comparable clinical syndromes. So it would be very difficult if you have a very specific, so for example, an anti-tau monoclonal, in order to assess its whether it is helpful in frontotemporal dementia, because you would only theoretically be only able to target or to help parts of these people, because others have other underlying pathology. When considering this sigma-1 receptor mode of action, which appears to be helpful across disease types, this would be perfect for frontotemporal dementia. So I would love to see if the company decides to do a drug trial there.
Perfect. Thank you. And the last question is, basically a roll-up. Do you have any other parting thoughts on Anavex's Alzheimer's data, the European Alzheimer therapeutic landscape, or future prospects for Anavex? Anything that you'd like to just close out with?
Yes, I'm doing Alzheimer's research for over 20 years now, and I really love to see that after a very promising beginning with the anti-dementing drugs I've been part of, it has been really hard time for around 20 years, seeing promising new compounds which did not achieve to show some clinical meaningful results in clinical trials. And now I love to be in this situation that it appears that several different compounds came up and new modes of action, so that all this research over the decades have resulted in some benefit that now hopefully can be brought to patients. And I would love to be in this situation to have in the future and the next year, hopefully a bunch of efficacious treatments to choose from in order to help patients with dementia showing up here. So I'm looking very forward to the next years, and hopefully we stay in contact. And also from my side, thank you very much for taking the time and having the county and the interest in everything I love to say, despite the fact that me as a psychiatrist tending to having very exhaustive answers instead of short answers you probably have hoped for. So again, thank you for being here. Enjoy your weekend.